Alright, we'll be looking at a new topic, cell signaling, right? We'll be looking at the stages of cell signaling. But before I begin, right, if you look at the learning outcome of this topic, there are two examples of signaling pathways involving insulin and glucagon, which you need to know as examples. However, right, for this topic, a lot of times in the A levels, in school exams, you can be tested on signaling pathways involving other signaling molecules, right? That's not insulin or glucagon, right? We can show you something you have not seen before. We can show you a signaling pathway that you have not seen before, and you're expected to be able to make sense of all these specific case studies and specific examples, even though you have not seen them before. All right, so that makes the general stages of cell signaling that we are going to cover here actually quite important, because when you are looking at a specific example, you are going to apply the general stages, and with the specific details given, then understand what's happening, right? So the general principles, the general stages of cell signaling is actually quite important for you to tackle all these case studies, application-based questions, all right? Alright, so having said the importance of this part of the lesson, we'll begin. Right, so in a general signaling pathway with any signaling molecules, there are usually three stages to cell signaling. Firstly, we have the ligand-receptor interaction, which is also sometimes known as signal reception. Then we have the signal transduction pathway, which happens inside the cell. And lastly, the cellular responses that are triggered, right, by the signaling pathway inside the cell. So we'll be looking at the ligand-receptor interaction first. Right, so before we look at the interaction, the process, let's look at what is a ligand. Right, so a ligand, right, is basically something that binds to the receptor. Right, so last year in when you learned membrane, right, you learned that there are different membrane proteins. Some are receptors, right, for signaling, for cell communication. All right, so here we would have something that binds to the receptor, and that's called a ligand. Right, so because a ligand is involved in signaling, right, so we also call the ligand a signaling molecule. And in the examples, right, in the syllabus, we have insulin and glucagon as signaling molecules, and insulin and glucagon are also known as hormones. Right, so in this topic, sometimes we will use the word hormones. Sometimes we use the word signaling molecule. So signaling molecule is a bit more broad, where hormones are a bit more specific to a type of signaling molecule. And then signaling molecule itself is a bit more broad. Or sometimes we will use the word ligand, right, when we are talking about the binding to the receptor. All right, so different words, different terms, which really mean the same thing. All right, so don't get confused if suddenly you hear different terms. Okay, so before we move on, just a bit more on what is a hormone, right, when we're looking at what is a ligand. Right, so again, in the syllabus, right, the examples given are insulin and glucagon as the ligand, as the signaling molecules, and they are both considered as hormones. So hormones are signaling molecules, right, that are secreted by cells in the endocrine glands directly into the blood. Right, so insulin, of course, is secreted by the pancreas, right, glucagon also by the pancreas, by different cells in the pancreas. So the pancreas would be the example of an endocrine gland. All right. So there are different types of hormones. Insulin and glucagon are both proteins, so they are both protein hormones. Most hormones are made up of proteins. There are some that are made up of lipids or steroids, when you learn about steroids, cholesterol last year when you learned lipids. Okay, examples of steroid hormones that are lipid-based would be your sex hormones, like testosterone and estrogen that you have learned in secondary school. Then there are another class of hormones made up of small molecules known as amines. All right, but most hormones are proteins, and again, the examples you need to know, insulin and glucagon, are both protein-based. Right, so for the ligand, right, there is going to be specific interaction, specific binding to the receptor. because on the receptor, there will be a part of the receptor known as the ligand binding site that have a specific shape and sometimes specific charge as well, right, to the ligand, to the signaling molecule. But at least complementary shape, if not charge as well. Okay? Right, so before I move on, right, ligand binding site is a term, because from experience, most students call every site on any protein the active site, right? But active site is a term for enzymes. Receptors are not enzymes, right? So you shouldn't be calling whatever site that's on the receptor the active site, right? It is called the ligand binding site, which describes its function very well, right? It is for the binding of the ligand. Right, so a very clear term that you should be using. Okay, so obviously a particular ligand, a particular signaling molecule, insulin for example, is going to bind to the specific receptor that contains the specific ligand binding site for insulin, right, for example. Right, so the signaling is going to involve cells that have the particular specific receptor for the particular signaling molecule, right? So again, for insulin, right, not all cells express the insulin receptor, right? You learned the control of gene expression last year also, right? So not all cells express all the proteins, right? So only certain cells, namely our muscle cells and liver cells, for example, right, express the insulin receptor. So insulin only affects the muscle cells and liver cells. Right, so there's the specificity of the action of the ligand depending on which cell contains the specific receptor that contains the specific ligand binding site, right, for the particular ligand, for the particular signaling molecule. Okay, so again, before we look at the process and what's going to happen in the interaction, after looking at ligand, we're going to look at what are receptors. Right, so receptors, again, you learned last year from the structure of membrane, right, that there are membrane proteins which function as receptors. So those are the cell surface receptors. There are other types of receptors that are not on the cell surface, but they are not really in the syllabus, right, but we are showing you just for info. Okay, so we focus on the cell surface receptors. So cell surface receptors are found on the cell surface membrane, right? The ligand binding site would be found outside the cell, or is found on the extracellular domain or extracellular part of the receptor. Right, so they would allow binding of the ligand, and most of the ligands that bind to the cell surface receptors are hydrophilic, water soluble, usually too large to enter the cell, right, through the cell surface membrane. That's why they are stuck outside the cell, and therefore they bind to the ligand binding site that are found outside the cell, right, in the extracellular domain of the receptor. So the examples that we have, right, with glucagon and insulin in the syllabus, they are both protein hormones, right? So they are both water-soluble, hydrophilic, large. So both of them do not enter the cell, but they bind to the cell surface receptors. Right? We'll learn more about the cell surface receptors for insulin glucagon in a later video, right? Now the intracellular receptors, we still going to introduce, even though it's not in syllabus, right? So they are found inside the cell, right? So logically, the thing that can bind to the intracellular receptors inside the cell must be able to enter the cell, right? So what kind of hormones, what kind of signaling molecules can enter the cell, right? Remember, we have the steroid hormones, right? The lipid-based hormones, right? So those will be able to enter the cell to bind to the intracellular receptors, right? So your testosterone, your estrogen actually enter the target cells, right? So a bit different from your glucagon and insulin. But anyway, this is not in syllabus. So this is just a slide and a picture to show what we mentioned just now with the cell surface receptors for the protein-based hydrophilic hormones, as well as the intracellular receptors for the steroid lipid-based hydrophobic hormones that can enter the cell. Right, so I should just move on. So this is a picture of a G protein linked or G protein coupled receptor, GPCR in this slide. It's called G protein linked receptor also, right? That's the term we use in the notes. Okay, so there's, you can see a G protein. The G protein is the, if you can see my cursor, G protein is this one, this whole thing, yeah? It's a G protein. But just a picture to show, right? We will talk more about the G proteining receptor in a later video lah. Right, this is tyrosine kinase receptor. Well, just again a picture to show you what the receptor looks like. This is the third example of the extracellular receptor, or the cell surface receptor rather. Right, this is the ion channel receptor. But we will not be looking at ion channel receptor in details because that's not involved in the specific examples that you need to know, right? Not involved in insulin or glucagon. So now we move on to the effect of ligand receptor interaction. So when the ligand binds to the receptor, this is like any protein, because receptor is a protein, right? So if you do not already know or do not already conclude for yourself, after learning so many proteins last year, whenever something binds to a protein, there is going to be a conformation change to the protein. All right, so it's the same for a receptor protein. So when the ligand or signaling molecule binds to the ligand binding site on the receptor, it triggers a conformation change which activates the receptor. So all receptors would undergo this conformation change. Some receptors, right, are made up of more than one subunits. So the ligand binding also causes the subunits to come together, right, which will then have a conformation change and activate the receptor. Right, that's for some receptors, but all receptors would have a conformation change with or without that aggregation of subunits when the ligand binds, right, and that activates the receptor. Once activated, right, the receptor is a transmembrane protein, right, actually, as you have learned last year. Right, so a transmembrane protein means it spans across the entire width of the membrane, right, and there is a extracellular domain, which just how we mentioned, with the ligand binding site that's outside the cell. Inside the cell, there's also a part of the protein known as the cytoplasmic domain or intracellular domain. Right, so that part of the receptor, right, after activation and conformation change of the receptor, would also be activated for binding of other molecules, other proteins inside the cell. Right, and when a cytoplasmic protein, we call these relay proteins. When cytoplasmic relay proteins bind to the activated receptor at the cytoplasmic or intracellular domain, right, the cytoplasmic relay proteins themselves get activated. Right, and then they go on to the next stage of cell signaling, which is signal transduction. Right, so what you can see here is, right, is these are especially your protein hormones, is how you mentioned, right? They are too large, too hydrophilic to enter the cell. Right? So if they don't enter the cell, right, how can they cause changes inside the cell, right? So this is where the receptor is important. The signaling molecule, the ligand, without entering the cell, right, just by binding to the receptor, right, causes activation, a conformation change of the receptor, which results in an activation of a relay protein inside the cell, right? So in this case, the signal, which is a very vague idea, the signal, right, is passed into the cell without the actual signaling molecule entering the cell, right? The signaling molecule stays outside the cell but can activate a protein, a relay molecule inside the cell, right, via the conformation change and activation of the receptor. Right, so this is the importance of signal reception or ligand receptor interaction. Right, it helps bring the signal into the cell without the signaling molecule entering the cell, because it can't. All right, so that's the importance of this stage of cell signaling.